Interphase. The cell grows and carries out normal cell processes, and DNA replicates.
Prophase, the first stage of mitosis, during which a dividing cell spends the most time. In this stage, the cell's chromatin tightens or condenses into chromosomes. In prophase, the chromosomes are shaped like an X. At this point, each chromosome is a single structure that contains the genetic material that was replicated in interphase. Each half of this X is called a sister chromatid. Sister chromatids are structures that contain identical copies of DNA. The structure at the center of the chromosome, where the sister chromatids are attached, is called the centromere. This structure is important because it ensures that a complete copy of the replicated DNA will become part of the daughter cells at the end of the cell cycle.
As prophase continues, the nucleolus seems to disappear. Microtubulous structures called spindle fibers form in the cytoplasm. In animal cells and most protist cells, another pair of microtubulous structures, called centrioles, migrates to the ends, or poles, of the cell. Coming out on the centrioles are yet another type of microtubulus called Astor fibers, which have an star-like appearance. The whole structure, including the spindle fibers, centrioles, and sister fibers, is called the spindle apparatus. The spindle apparatus is important in moving and organizing the chromosomes before cell division. Centrioles are not part of the spindle apparatus in plant cells. Only spindle fibers are present. Near the end of prophase, the nuclear envelope seems to disappear. The spindle fibers attach to the sister chromatids of each chromosome on both sides of the centromere and then attach to opposite poles of the cell. This arrangement ensures that each new cell receives one complete copy of the DNA.
Metaphase. During the second stage of mitosis, metaphase, the sister chromatids are pulled by motor proteins along the spindle apparatus toward the center of the cell and line up in the middle or equator of the cell. Metaphase is one of the shortest stages of mitosis, but when completed successfully, it ensures that the new cells have accurate copies of the chromosomes. Anaphase. The chromatids are pulled apart during anaphase, the third stage of mitosis. In anaphase, the microtubules of the spindle apparatus begin to shorten. This shortening pulls at the centromere of each sister chromatid, causing the sister chromatids to separate into two identical chromosomes. All of the sister chromatids separate simultaneously, although the exact mechanism that controls this is unknown. At the end of anaphase, the microtubules, with the help of motor proteins, move the chromosomes toward the poles of the cell.
Telophase. The last stage of mitosis is called telophase. Telophase is the stage of mitosis during which the chromosomes arrive at the poles of the cell and begin to relax or decondense. Two new nuclear membranes begin to form and the nuclei reappear. The spindle apparatus disassembles and some of the microtubulus are recycled by the cell to build various parts of the cytoskeleton. Although the first stage of mitosis are now complete and the nuclear material is divided, the process of the cell division is not yet complete.